Hi, good morning, everyone. Today, um, I will talk about 3D reconstruction of neuron using the zero block phase SEM. So I'm from NIH, National Institute on Drug Abuse, RP Confocal, and the electron microscopy core. Basically, today, I want to share my experience. We did not publish the data from this set, but I have a lot of kind of things I want to share with you, especially for people who are doing neuroscience, the neuron and work on for the mouse, rat, brains. So our institute work, study for the drug abuse. Drug abuse exert their initial reinforcing efforts by activating reward circuits in the brain. So basically for the reward system usually work for the dopamine systems and from one brain area, mostly from VTA, the ventral tegmental area, and two different brain areas. So mainly for reward, but now the behavior include aversion, pain, positive reinforcement, and negative reinforcement, decision making, and social behavior, and a lot of different behaviors. So that is for the VTA. So VTA is heterogeneous. So the previously beginning mainly focused on dopamine neuron, and then the GABA neuron, neurons for the inhibitory function, and the glutamate neuron for excitatory functions. And in recent years, scientists frequently reported for the combinatorial neurons, including dopamine neuron peptides, or dopamine GABA, or dopamine glutamate, or glutamate GABA. So our group discovered this type of neuron, glutamate GABA, but we had we need to answer the scientific question about is that excitatory or inhibitory. So that's why now I want to share why we are doing 3D reconstruction. For people who are not who has no background for the neuroscience, I want to briefly talk about introduce some basic background about neuron. So a neuron has three main parts: a cell body, and or soma you call, and dendrites, and single axons, and the axon has axon terminal. So for most neurons receive signals where the dendrites dendrite or soma, and send out signals down the axon. So as the majority of synapse, signals cross from axon of one neuron to a dendrite or soma of another neuron. So a synapse is a structure that permits a neuron to pass an electrical or chemical signal to another neuron. There are two types of synapse. Asymmetric synapse has a thick PSD. It's a postsynaptic density, and symmetric synapse has a similar with pre and post density. So asymmetric synapse usually is excitatory in function, and symmetric synapse is inhibitory in function. By detecting of the VGluT2 mRNA, GAD mRNA or VGAD mRNA using RScope, we demonstrate the distribution of VGluT2 only, VGAD only, or VGluT2 VGAD dual neuron. So that's the we found. Previously people thought, oh, GABA neuron only have has VGluT2, and no, glutamate neuron has VGluT2, and GABA neuron has VGAD. But we found, interestingly, there's VGluT2 and VGAD co-expressed in this neuron. And to determine whether this lateral habenula area from basically we have some data for the behavior and E-phase data, and then we found GABA and glutamate regulated by each other, but we did not know by that time there are neurons in the VTA containing VGluT2 and VGAD. But first we found in the terminal area by immuno TEM, we found we did a double immunolabeling by as you can see, the DAV staining seen as the dark diffuse material, and also we use the gold particle to detect another molecule. So in this study, we found some terminal has only VGluT2 and some terminal has only VGAD, and we did see terminals containing both VGluT2 seen as dark diffuse material and VGAD seen as gold particles. And this terminal contain asymmetric synapse and symmetric synapse. Then we deliver independent AAV including channel-adopsin 2 mCherry into the VTA, and then we study the lateral habenula area. From our confocal data and 3D reconstruction, clearly we can see the mCherry, this axon and axon terminal, especially in the axon terminal, co-expressed VGluT2 and VGAD. We detected the expression of the VGAD seen as a gold particle co-expressed with the mCherry, basically from glutamate neuron from VTA. And from serial section, we can observe asymmetric synapse and symmetric synapse. And further, we isolated the vesicles from the lateral habenula area, and then we did the immunogold detection for the VGluT2 and VGAD. We found these two, like this, we use a different size of gold particle, this 18 nanometer and this 12 nanometer. And we don't see these two sides of the gold particle located in the same vesicles. So that means this in the lateral habenula, VGluT2 and VGAD are in the different pooled vesicles. So, and then we concluded that a single glutamate GABA axon terminals make independent asymmetric and symmetric synapses on the lateral habenula. But however, we were not sure whether these two dendrites are from the same neuron or different neuron, because we did the serial section by TM, but there's limited serial sections. We cannot answer the question like, do drug overabuse alter the delicate balance between excitation and inhibition. So here is a diagram showing the limited serial TM image cannot capture everything. So that's why you can see here, you see this, just like the dendrites. We found that they synapse different dendrites. We don't know they connect by each other or separate. So that's why we started to do 3D reconstruction. So we thought 3D reconstruction is the only technology that can effectively confirm this phenomenon. So here's the workflow. Basically, we got this protocol from once we purchase, after we get this this review, Gatan's review, and that's Sigma VP system, and we got the protocol recommended from ZAS, and later on we modified the protocol from the Tom Derek. So we perfuse the animal, and we section. We at beginning we did 100 micrometer, and eventually we found the 250 micrometer thickness better. And then the the heavy metal staining and the dehydration and infiltration. After the polymerization, we mount the sample on the spin by the conductive glue and trim the sample. We put the sample in the chamber where you can see the diamond knife, and we also use this focal charge compensation. That's after a couple of years we tried. So, and then we did the serial image collection. So here's our the microscope Melanie already showed. So we have almost the same, but maybe that's microscope is different, but the 3-view chamber is the same. Inside, there's a diamond knife and can automatically cut from 15 nanometer to 200 nanometer. And usually we cut 17, I think we tried 50, also 70 nanometer, and the fresh face of the block is scanned and imaged with each cut. So the resolution, I think the resolution is pretty good. So here's all details for the image accumulation. We usually use 4 or 5 nanometer by 4 or 5 nanometer pixel, and the thickness, I think we eventually we use 70 nanometer. And after we collected all the image, we use Dragonfly to process image segmentation. First, we use Amira software to align the image, and then we process for the segmentation. So here's our result. So we found, for this type of, I mean, we did not label the neuron type because we tried to label with gold particle, but we had charging problems, but we know in the lateral habenula, 70% of the terminals has co-expressed VGluT2 and VGAT. But we're still not sure, but we know we found some terminals has symmetric and asymmetric synapse, and from 3D reconstruction, we found these dendrites are from different neurons. So here's our movie, 2D and 3D movie. We can show this axon terminals makes asymmetric synapse and symmetric synapse on different dendrite and different neurons. However, we still cannot completely answer the question. We know that we don't know this type of neuron. We only can guess. So that's why there is a limitation for all research for this protocol and technique. So that's why. So now I want to tell like my experience about this cerebral phase SEM in our place, because we are doing the neuroscience, not like other place. So we have to know neuron type, even though for the neuronal network, we need to know different. Maybe we need to label at least two or three different neurons. We know this neuronal network. So we had a problem for the immunolabeling. And also we had problem of to balance the dose. So like the exposure time or pixel size and the work distance. And we also had the problem for the serial image quality continuity. So at beginning, we could image and section several hundred of the serial image, but I don't know, after years, we had a problem for, at beginning, the serial section is good. And then after maybe 20, 30 serial image, and then they get like either burning or the dirt diamond knife problem. But anyway, we did not have a very good luck at the late stage of this technique. And also the FCC, the percentage. So at beginning, like we use 10, 15 to 20, but later we need to increase. But once we increase, it affect the resolution. Anyways, for our study, we couldn't answer what type of neuron. That's why we decide we need to move to another technology to solve our scientific question here. That's why 3 years ago, we attended a workshop organized by Naomi from Max Planck of Florida Institute. So after that, we purchased all equipment, MRC, Autotome, and we are doing correlative light and electron microscopy. So I don't have time to present the details, but I want only show we successfully get this method work, and we can do the correlate the low magnification from confocal image and EM image, and this high magnification we can label the axon terminals by different channels like UIP or and then we detect the VGLUT2 with one channel we get with another channel, and then we merge. And for the serial image for this SEM image, we can correlate exactly same place for this terminal. We know the R VGLUT2 we get co-expressed terminal and make a symmetric and asymmetric synapse to the different neurons. And here's the zoom in to show this. And here's the after segmentation, we can see these terminals. Here's the 2D and 3D video to better show this terminal. To show this this terminal make a symmetric and asymmetric to different dendrite or dendritic spines. And so I would like to thank Dr. Marcela Morales, who is the course. We have several courses here, and she's a director, fully support all EM core. And I also want to thank all core staff. Kevin learned this, Kevin did the 3D for the clamp and also for the clamp and learn this new technology. And Alex support the data analysis, and Aaron did a lot of immuno TM. And also I want to thank Jeanette, who is the scientific program coordinator, for helping the new equipment purchase. And Lam is the scientific program analyst. And also I want to thank Shiliang, who is the summer student. She first started to analyze this the using Dragonfly. She contacted Alice group from NYU. We got this, we learned this, and now we are doing better. And I also want to thank NIDA LP for the fund, and I would like to thank all the collaborators, and especially I want to thank the BioEM group. I got the benefit. I learned a lot, and Ruqing always help. We are all in the Baltimore, and we got help from Alice and also Naomi, and a lot of other colleagues in this group. So thank you all. I would like to take any questions.
Yeah, if you have questions, you can raise your hands or go through the Q&A. And I also want to point out that in the later part of the later sessions, we do have a correlative microscopy session that we plan to run. And there's also a array tomography session we plan to run. Maybe we'll bring Shiliang back to talk about his correlative microscopy. Trace, please go ahead, ask your questions.
Yeah, very nice talk, Shiliang.
Thanks.
Thank you. Can you, so you mentioned Dragonfly software for your renderings early on, and those are very nice renderings. Can you speak a little bit to that software as far as ease of use and if it's applicable? Like, for instance, did you use it for your autotome wafer data at the end also?
Yeah, we are using, so we tried a different software. Before we contact Alice, we were using MRIs, and there was some problem for the segmentation, and so we have good luck for the Confocal 3D because there are not many sections, but for the EM data, I would like, we try different software. We think Dragonfly is very good for this segmentation. But before using the, put in the Dragonfly, we need to align the serial image very well. So we usually use a mirror software to align it and then do the segmentation. Yeah, this is software. It's free for all this non-commercial, and we pay very little. It's very, we pay only like 2,000 something for the every year the service contract. So this support technically. So now for the CLAM, we are also using this software for this 3D reconstruction. It's very, very powerful.
Good, thanks.
Alice, you go ahead with your questions.
Very nice talk, Shiliang. Well, one question, maybe I'm not very clear for the last experiment you show using the clamp to do in the autumn section correlation. So did you, so all you, I guess all your endogenous expressed protein, you image them with the vibraton section, then you directly embedding the vibraton section using the OTO method, or you use a different embedding reason so that when you do the series section, you can image the signal. In the meantime, use SEM. You image the light microscopy signal. Then in the meantime, use SEM to image the high resolution signal. What did you use?
So for the thanks, Alice. You have a lot for to make our project move forward. So for this clamp, it's different from the 3D the previous we did. So we just perfusion, we just regular perfusion, not a special perfuse like heavy metal. We don't need that. But after perfusion, we vibraton section, we process immunostaining. So we also have not all endogenous. So we have GFP. We need to tag the neuron, the VGluT2 VGAT double neuron. So we need to inject the virus, and then we follow this GFP, and then we do the immunostaining primary antibody and secondary antibody, so after
so then you have to use a triton to treat the vibraton section before you do the immunolabeling with the fluorophore, right?
No, we don't use triton.
So basically, you can just directly do immunolabeling on the vibraton section.
No. So vibratomes. We just process just like a immuno TM. We process, but we don't use triton. We use saponin. Okay. So help the penetration. So usually, for penetration is not a problem in our case if we use saponin in the blocking buffer, and then we collect.
Do you know how deep the signal can penetrate? And I mean, for fluorophore, maybe it's really easy. I'm not sure if you have ever tried to think about the use of, use the fluorophore or nano, fluoro nano gold kind of thing. Then you have both signal.
We tried. We tried the nano gold. They have fluoride from a nanoprobe. It's not. Not good in our place. We can see a little bit signal for the nanogold for the surface, but usually we need to trim and cut, and it's hard to follow. And eventually at beginning we ordered the nanogold with the fluorescence, and then we found it did not work very well. So we just use the fluorescence, the regular primary antibody, secondary antibody, and then we collect the serial image. So we cut, now we cut 100 micrometer. So the penetration for both sides is okay. Only in the middle a little bit, it's okay. We in the 3D reconstruction, we can, for the segmentation, we can track, we know what type of neuron. So the terminal, we mainly focus on the terminals and also some cell, but in different project, we need to know the type of the post synaptic neuron. So sometimes like this project, we cannot do many channels. So we have a GFP, VGluT2, VGAT, and also the nucleus. So we know where's the cells. We have four channels. And then so after
basically you can use a confocal to do this tag to get all three.
Yeah, we did that.
Light microscopy imaging first, right?
Yeah. In the confocal you can always do low mag and high mag. We use a 63, even though zoom 2, and then we get the Z stack, because eventually we need the Z stack to correlate for Z SEM image. So yeah.
Very good. Thank you.
Okay, thanks.
Shiliang, there's a question in the Q&A. David, do you want to ask the question yourself? Don't know if you have your mic on. David, by David Shatlet.
Oh, so the question is why we use a mirror and not directly use Dragonfly. I think Alex tried from our group tried to use the Dragonfly align that. I think did not align very well. That's why Alex is here. So Alex, I don't know. Oh, he's not in the panel list. So we had a problem directly use if we directly use the Dragonfly to align, there's a problem and it's very time consuming. So we just used, we had a mirror 3D software. It's easy for us after aligned, and then we put all this directly to the Dragonfly. Maybe if you know very well for the Dragonfly how to align, we would like to learn also, because that's a way last summer when the summer student was here, we had this trouble. We found, oh, a mirror worked very well, and then after that we use a Dragonfly.